in Science and the MS program in Environmental Science and Management at the ISU. Dr. Sato earned his PhD in Environmental Engineering from the University of Iowa. With a broad background in environmental research and practice, Dr. Sato focuses on the physical, chemical, and biological treatment of water and wastewater, surface and subsurface water quality, paint and platform renewable energy development. He was the president of the JSPS Fellowship from the Japan Society from the Promotion of Science from 2002 to 2003, and was a Fulbright for 2012. Teaching and conducting research at the Tripura University, Narayan campus in Pokhara, Nepal. He was also invited visiting professor at the Graduate School of Engineering, recent tenure of environmental quality management at the Idaho University in Japan in 2014. 2017, Dr. Sato was awarded the Chinese Academy of Sciences President's International Fellowship Initiative, ISB, fellowship under the auspices of the Institute of Urban Environment in Xiamen, China. Currently, he serves as a guest editor of the Journal of Energies from the special issue Microbial Fuel Cells, 3rd edition. He has also served as a guest editor for Applied Science, Energies, and Membranes, and as a founding editor for the International Journal of Environmental Engineering from 2009 to 2024. And the International Journal of Environment and Waste Management from 2008 to 2014. You can continue on, Mr. Sato.
Thank you. Because the voice seems sort of scattering around, so I will speak very slowly. Good afternoon, everyone. My name is Chikashi Soto from the Department of Civil and Environmental Engineering at Idaho State University in the United States. It is truly an honor to be invited as a guest speaker at your distinctive institution. I'd like to express my sincere gratitude to the organizers for giving me the opportunity to share my research work and for setting up such an important event. Without further ado, let's learn together. The title of my talk today is Microbial Fuel Cell with Ceramic Membrane Separator for nutrient recovery from potato processed wastewater. Idaho is well known for potato production, the #1 state for potato production. My co-author, Ghazale Alikaram, is a master's student currently working for a master's degree. Wilkins Apron, he was a really good mentor for Ghazale when he was here at ISU from a summer 2022 to winter 2023. This is outline of my talk. I start from a introduction with brief background and goals and objectives. Then I'd like to focus on microbial fuel cell combined with hydroponics, we call MFC hype system. Then I'd like to draw conclusions, then future research needs, and I'll then conclude my address, addressing any questions you may have. So as the world population grows, the urbanization continues, and global climate change progresses, the demand for energy, water, and food surely rise, accompanied by the need for the retreatment and reuse of wastewater. So this study focuses on generation of electricity, wastewater treatment, and food production. The goal of this research is to develop a sustainable energy, water, food supply system capable of simultaneously generating electricity, producing value-added product, in this case, edible plant, and treating wastewater using potato process wastewater as a renewable source of energy and nutrients. The broad objective is to explore microbial fuel cell, MFC, hydroponic system for sustainable energy, water, food supply system. The specific objectives: one, to recover energy from wastewater as electric power. Two, to recover nutrients from wastewater as value-added product, which is edible plant. The third, to remove organics as COD from wastewater. Over the last several decades, bioelectrochemical processes have drawn considerable attention as they can be put into operation with low or near zero energy input. So microbial fuel cells are devices that convert chemical energy to electrical energy using microorganisms, the special microorganisms, so-called exoelectrogenic or electroactive bacteria, EAB for short. We use bacteria as biocatalyst. The MFC are structurally classified into two types: dual chamber or two-chamber microbial fuel cell and single chamber MFC. So this is two-chamber or dual chamber microbial fuel cell. This side, this is anode side, this is anode chamber. This is cathode chamber interfaced by PEM, proton exchange membrane. In the anode chamber, wastewater, which contains organics, the organics is oxidized by EAB bacteria, release electron and proton, and carbon is converted to carbon dioxide. The electron flow through external circuit to the cathode, where oxidizing agent, in this case oxygen, is reduced to water, accepting electron and proton. Now, this is single chamber MFC. The single chamber MFC, the cathode chamber is removed. Instead, the cathode is directly attached to the surface of anode chamber. So electron is released by EAB. So electron flows through external circuit, then goes to the cathode, because this is called air cathode. So the cathode is directly exposed to the atmosphere. Therefore, oxygen is abundant. So oxygen receives the proton and electron to form water. So this is single chamber MFC. Now, the potential applications of MFC, the originally idea to start the generation of electricity using organic material. Then further extend this to wastewater treatment applications. So removal of biodegradable organics and nutrients. Then further extend this to bioremediation, treating toxic waste or removal of recalcitrant organics and heavy metals. Then further extended to biosensors, the environmental sensors, or even biomedical applications. So this study focusing on wastewater treatment, then nutrient contained in wastewater is recovered using hydroculture system. Now, this is the system which has been published by a number of researchers. This system combining constructed wetland, CW, constructed wetland, and MA microbial fuel cell combined, and HP hydroponic system combined with microbial fuel cell. So for example, this system, this is proposed by Liu et al. 2013. The column with filled with gravel, this is gravel, then granular activated carbon is inserted between. Then granular carbon also added on the top. Then this portion become anode because there is no oxygen. Then this portion on the surface, this serves as a cathode. This anode and the cathode connect this to generate electricity. Plant was placed on the top. Then wastewater was fed from the bottom. Then wastewater flow upward, then overflow, then released from the top. Now, this system further modified in here. This was published in 2019, same author. Now, wastewater is fed from the top, then flows down, then goes up, then released from the top. Now, this section is anode. This section served as a cathode. The anode and the cathode were connected to generate electricity. This system, this is proposed by Kuhlman et al. 2020. This is a column. Then in the middle of the column, ceramics plate is placed right here. Then anode, this is anode. This is a cathode. So now you have an anode chamber and the cathode chamber. On the cathode section, the plant was placed. Then wastewater fed from the bottom because ceramic plate is almost impermeable. Therefore, bypass line was made. So wastewater flow from the bottom to move to upwards to the cathode zone. There was a plant was growing. Okay. Now, then this is similar to two-chamber microbial fuel cell. Now this one, there is no the ceramic plate, so only this chamber. This is like a more single-chamber microbial fuel cell. So this line right here. This is the anode. The cathode was placed near the top. That's where the oxygen is available. Then wastewater flow from the bottom to top. This system, this is the anaerobic biofilter was placed to treat low urban wastewater. The treated wastewater was fed to the hydroponic system. Anode was placed the bottom of the tank. Then cathode was placed near the root zone, that's where oxygen is produced. Then cathode and anode are connected to produce electricity. Now, all these processes has one thing common. That is the wastewater directly contact with the plant. So if you grow edible plant, you have some problems. So here, so the world, the prevention, expansion of numerous the waterborne diseases across the world raises serious concerns. And there is a need for protecting our food from contaminated water. So to address these concerns, we explored the new MFC hydroponic system. We use Arium tuberosum, commonly known as Garlic chives and Chinese chives, and we call Nira in Japan. We use this plant as hydroponic plant. This plant is tough, fibrous, edible, perennial plant. It is easy to grow in different conditions. It grows even Idaho. And it is rich in antioxidants, which has antimicrobial activity. This is a conceptual model. The scale, this is not scaled. And this is microbial fuel cell, which is submerged in hydroponic the system. The wastewater is fed into microbial fuel cell. This is anode. That's where the bacteria grows to oxidize organics. Now, organics is converted to hydrogen or proton and carbon dioxide and releasing electron. Electron flows through anode to cathode. This is the cathode. Then between cathode and the anode chamber, this is a ceramic plate is placed. Now, then the carbon dioxide produced by biological activity, because the microbial fuel cell is a closed system. There is no way to escape. Therefore, carbon dioxide dissolves into wastewater to become either carbonate or bicarbonate. Phosphate, there are plenty of phosphate in wastewater. Ammonia, also plenty of ammonia, and also organic nitrogen is hydrolyzed to form ammonia. So ammonia diffuses through from microbial fuel cells to hydroponic water. Phosphate also diffuses through. Bicarbonate, carbonate also diffuse through. Electron is taken by oxygen and hydrogen combined to become water. Now, all these nutrients will be taken by the plant. So these are the concepts. Now, these are our MFC design. We use bamboo charcoal. This is about 4 plates of bamboo charcoal because it's environmentally friendly. So this is for bamboo charcoal. This is the anode. Then this is the open hole right here. This is air cathode exposed to atmosphere. So this is the diagram right here. So this is bamboo charcoal anode. The bacteria grow on the surface. Then organic matter will oxidize to form proton and carbon dioxide. Electron flows through external circuit to the cathode where oxygen is reduced to water, accepting electron and proton. So this is our microbial fuel cell. This is a scanning electron microscope image of bamboo charcoal. As you see, the bamboo charcoal has rough surface, which provides easy attachment site for bacterial growth, and also provide large surface area, which allows large number of bacteria to grow. That means more electrons are transferred to the anode. The separators, the desired characteristics of the separators are as follows. Which has high ion transport, because phosphate and nitrate and ammonia have to pass through. High biocompatibility, which means non-toxic. High chemical and thermal stability, which is corrosion resistance. High mechanical strength, environmentally friendly, which means safely disposed of after its life, readily available at low cost. Now, ceramic membrane have many of these characteristics, almost all these characteristics. High solute flux, which means high transport. The ceramics have high bacterial resistance and biocompatibility. Ceramics have high chemical, mechanical, and thermal stability, high durability, easy to clean, and has long work lifetime, lasts a long time. Environmentally friendly and low cost and readily available, as it is made of clay. Clay is everywhere. Now, specific requirements for the MFC hype systems are, so that it must allow diffusion of nutrients and carbon species from the MFC to hydroponic water. And also, it must retain substances other than nutrients and carbon species, especially pathogens within MFC. So all the pathogens, bacteria, other stuff will be retained in microbial fuel cell, allowing nutrient and carbon species to the hydroponic water. This is the SEM scanning electron microscope image of ceramics. This is 221 times magnification. This is 800 times magnification. So if you see here, there are small holes throughout spread around. This is the largest hole we can see here. This is about 20 micrometers. However, this hole does not penetrate the other side. The thickness of this ceramic is about 4 millimeters. So this is only surface. So this is not a big concern. This is MFC height setting. This is a microbial fuel cell submerged in hydroponic system. Then two sets of plant, the location of A. So location of A is 6 centimeters away from the MFC, this here. The B is located 16 centimeters from my MFC. So hypothesis is that if nutrient diffuse from the MFC, the plant located at A grow more than the plant located at B because the concentration of nutrient is high at the location of A than location of B. So this is a setting. We have a 3 sets, each almost identical. This is microbial fuel cell. This is plant location of A. This is closest to MFC. This is location B. Initially, we cut the plant to 8 centimeters. Then during the run, the covered with card box to prevent the growth of algae by preventing light. These are the conditions: temperature 22 degrees Celsius, the volume of MFC 520 milliliter, the volume, the effective volume of hydroponic vessel 2,500 milliliters, the depth of water 6.25 centimeter, operated by batch. Then we run 3 systems, 3 units simultaneously. These are the characteristics of potato wastewater, potato process wastewater. pH about 6.9, conductivity 64 millisiemens per centimeter, COD 2,670 milligram per liter. These are the nutrients: ammonium nitrate, phosphorus, potassium, then other constituents, sodium, magnesium, aluminum, so forth. This table shows the wastewater quality beginning and end of the run. The run lasted 49 days. The pH in the beginning, 6.8, 6.9. At the end, the pH increased to 8. So about 17% increase in pH. Conductivity, 64 millisiemens per centimeter, which is increased to 92.5, about 45% increase in conductivity. COD, in the beginning, 2,670 milligram per liter, which is reduced to 100 milligram per liter. So 96% reduction of COD. Ammonium nitrogen was 0.2 in the beginning, 0.4 at the end, so about 100% increase. This is due to ammonification of organic matter in the potato wastewater. Nitrate in the beginning, 0.38 milligram per liter, increased to 0.41 milligram per liter, about 34% increase. This is due to nitrification. Now, power output was calculated based using these equations. First, we use Ohm's law. The voltage, we measure the voltage, and we know external resistance. It's a load. So the voltage divided by external resistance gives current. So using this current, so current multiplied by voltage gives the power. Then power divided by cathode area. This gives, we call the power density. So unit of power density is milliwatt. This is power density. Unit we reported as milliwatt per square meter of cathode area. Now, so this is called the power curve. This is the maximum power density right here. This called polarization curve. Right? Now, so here we run 3 units. Then, this is a MFC alone. This is MFC hydroponic system with the plant. This is MFC hydroponic system without plant. So this is average MFC alone. The average power is 524 milliwatt per square centimeter of cathode area. This is MFC alone. Now, MFC combined with hydroponics with the plant produced the power, maximum power, 130 milliwatt per square centimeter. Now, MFC with hype, without plant, produce 99 milliwatt per square meter. Now, I summarized in this graph right here. So this is maximum power density. The MFC alone produced 524 milliwatt per square meter of power. This is MFC combined with hydroponics with plant produced 130 milliwatt. MFC hype without plant produced 99 milliwatt. Now, I calculated internal resistance, then shows right here. Internal resistance of microbial fuel cell alone is 114 ohms. The MFC and hype with plant internal resistance is 840. Then without plant, 900. So seems the plant reduces internal resistance. So 900 to 840. This affected the power output. So here, the without plant power output is 99. With plant, 130. Okay. Now, the big difference from to MFC alone, because the increase in internal resistance is due to ceramic. So one major disadvantage using ceramic is increase internal resistance. Now, this graph shows the change in the leaf length. So this is length of leaves. These two are round one, the MFC hydroponic 1. This is MFC hydroponic 2. This is MFC hydroponic 3. 3 continuous, in parallel runs, right? Now, this is control, hydroponic only, no microbial fuel cells. This is location A, this is B. This is A, this is B, this is A, this is B. So this is the following observations. The leaves in the MFC hype system were, on average, 61% taller compared to those of control, which means this without microbial fuel cell, with microbial fuel cell, 61% taller. the second observation, the leaves located at A, which is closer to the ceramic membrane, or MFC, are on average 16% taller than those grown at location B. So the preliminary conclusion is nutrients diffused from MFC to hydroponic water through the ceramic separator. Now this graph shows weight of leaves, and blue color represent wet weight. Wet weight. The brown color represent dry weight. Again, these are microbial fuel cell with submerged on hydroponic system. This is hydroponic only, no MFCs. So this is A, this is B. This is location A, this is location B, and A and B. So the observations are as follows. Wet weight of the leaves, this is blue color, in the MFC hype system was on average 189% heavier than those of the control plant. The dry weight of the leaves in the MFC hype system was on average 142% heavier than those of the control plant. Now, based on the locations A and B, wet weight of the leaves of the plant grown at site A are on average 50% heavier than those grown at site B. Well, based on dry weight, the dry weight of the leaves of the plant grown at site A are on average 38% heavier than those grown at site B. So preliminary conclusion is that nutrients diffused from the microbial fuel cell to hydroponic water through the ceramic separator. Now, these are the temporal concentration of nutrients. This green line is the hydroponic system with plant. This is with plant. The blue line is without plant. This is 3 line for the measurement of sodium. As you see, concentration increases with time. That means sodium is migrating from microbial fuel cell to hydroponic water. Another observation, because the concentration of sodium with the plant is lower than without plant. That means the plant taking up sodium. Now, this is potassium. The interesting thing is now concentration is coming down. About 1 or 2 weeks after, that means the potassium uptake rate by the plant is larger than the rate of diffusion of potassium from microbial fuel cell to the hydroponic system. That means the growth of the plant, A. tuberosum, was likely limited by potassium in the microbial fuel cell hydroponic system. Now, the same plot, this is phosphorus. and nitrate. Again, the phosphorus concentration is lower, it increases with time. That means phosphorus is diffused from microbial fuel cell to hydroponic system, and lower concentration compares to without the plant, and plant is taking up phosphorus. This is similar to nitrate. Nitrate concentration increases with time. That means nitrate or ammonia is released from microbial fuel cells to hydroponic system, and ammonia is oxidized to nitrate by nitrification. But nitrate is also taken by the plant. So these are the conclusions. The result of this study supported the concept that nutrients diffuse from the MFC to the hydroponic system, then where plant is cultivated. The nutrients are taken up or recovered by the plant. The organics in the wastewater can be removed in the MFC while electricity is generated. The concentration of organics in the wastewater was significantly reduced. We observed 96% reduction of COD in the MFC. Then, while electricity was produced with microbial fuel cell with hydroponic system, 130 milliwatt per square meter of the electricity or power was produced. Then, Arium tuberosum, which is edible plant, was produced. Then, this is additional conclusion is that the nutrient stayed in ionic form at the cathode, because the cathode is always wet, because it is in contact with hydroponic water, and the pH of the water does not increase significantly to cause formation and accumulation of precipitates around the cathode. Therefore, the cathode requires less frequent cleaning. So this is the added advantage. So the future research needs, the following question needs to be addressed. The first, was the power output by the MFC really increased by the plant? The preliminary result I showed shows yes, it did, but it needs to be confirmed. Second question, can the nutrient transport through the ceramic separator be enhanced by the increasing electric power? Again, the result we obtained showed yes, but still needs to be confirmed. The third question, can the plant growth be further enhanced by increasing nutrients and or inorganic carbon in the hydroponic system? These are the next agenda we wanted to investigate. So thank you for your attention, and I'd be happy to answer your questions. Any questions?
I have a question for you. Dr. Question. So, another member has the retain COVID-19.
I'm sorry, I cannot hear well. I think it sound is not really all the sound is scattering. Sorry, I cannot hear well.
Can you hear me well now?
Oh, now it's better. Yes.
Okay. So, Dr. Evgenia has a question for you. And it is that does ceramic membrane has the ability to retain the COVID-19 virus?
Now, that if I can hear correctly, your question is the ceramic membrane can retain other constituents. Is that your question?
Sorry, can you repeat it again, please?
Let me, because of the still have a problem, the ceramic membrane we have, the purpose is to diffuse the nutrients and carbon, which is bicarbonate and carbonate species through the two hydroponic system, but trying to retain any other materials, including bacteria, and because this is a potato waste material, there are a lot of other things in it. For example, like a starch, right? And protein. Sometimes there is lots of organic, high molecular weight compounds. So where everything is contained in the MFC, then because this is like more like anaerobic digester. The only difference is the methane is not produced. The only carbon dioxide is produced in the system. So hopefully everything is retained in microbial fuel cell. Only nutrients and carbonate species are diffused to hydroponic system. Because if I'm not addressing your questions, please send me email. I hope I can answer better because sound is not really transferring well.
Thank you so much, Mr. Keller.
Any other questions? Still, we may have a sound problem.
From our side or your side? Now we're going to look for the accident. Now, Mr. Faro, thank you so much.
Thank you very much.
We're going to send you later the announcement, Mr. Faro. Thank you very much for your patience and your time.